So today we're going to start with the life processes in living organisms, the part 2. Do not worry whether it is part 1 or part 2. The thing is that I wanted to start with the reproductive system, okay? So in this particular, video, we'll be dealing with reproduction, asexual and sexual reproduction, okay? Sexual reproduction only till plants, only up to plants I'll be dealing in this particular part. In the other part, we'll be dealing with the animals one. So let us first begin by, this I've not taught you about the cell division and all that. I'll be dealing when I complete this chapter. No, I'll come to cell division also. So I'll not discuss this can you recall one, because this is part 2. Above you will see part 1 is there here. We have this cell division. Okay, look here. Mitosis, prophase, anaphase, telophase. This one I'll tell you after we complete this one, because this is very easy. Okay, reproduction part is very easy. So we'll start with here only, asexual. Alright, let's start from here. So what is asexual reproduction? It is the process in which a new organism is formed. Okay, a new organism is formed, but there is no genetic recombination. Remember that in asexual there is no genetic recombination. Okay, no genetic recombination is there in asexual reproduction, and no involvement of gametes is also there. Okay, no involvement of gamete production takes place. Also, the same species is formed. A kind of, we give it a specific name, clone. A clone is formed. Okay, and as this reproduction, it will not involve any kind of gametes. So what will happen? Exact genetic similarity will be there. And it's a uniparental reproduction, which means only one parent is involved. Only one parent is involved. And since the gametes are not involved, the germ cells are not involved, so which kind of cell division? I hope you know this, mitosis and meiosis. The basic thing, like meiosis, it happens in somatic cells, and meiosis takes place in germ cells, that's the sperms and the ovum. Why is the mitosis takes place in somatic cells? Somatic cells are the body cells. So thereby here no germ cells are involved. So of course, which kind of cell division will be there? Mitotic cell division will be there. I already told you absence of genetic recombination is there. That's a drawback. But comparatively, asexual reproduction is quite fast than sexual reproduction. Okay, it is very fast. You must have heard of bacteria which can divide in about 20 minutes, right? I hope you all know about it. Now, let us come to unicellular organisms here. So now we are going to talk about the asexual reproduction in unicellular organisms. So in unicellular organisms, first we are going to, I'll just make a flow chart for you here. Okay, once we are done with the flow chart, it will be very clear. Just a second. Yeah. So here we go. Yeah, so I'm going to make a flowchart out of it. So here we do have it. Asexual reproduction. First process is fission. Okay, I'll explain fission here. Now in fission we have two types: binary fission and multiple fission. So this is I, okay, I. Multiple and binary. Now binary is again of two types. It can be transverse and it can be longitudinal. Let's first discuss only fission. Now remember here, in binary fission, if I talk about binary fission, so binary fission always takes place under favorable conditions. When plenty of food is available for the bacteria or any other whatever the organisms are in binary fission to divide. While multiple fission, it takes place under unfavorable conditions. Now here, the formation of cyst will take place. I'll tell you what cyst is. This formation will take place. Okay, this is clear, no? Now look, I'll tell you about binary fission in short, because we have already dealt with binary fission. We already know what binary and multiple fission are. In a very short manner, I'm going to tell you important things first. 1 minute. Now look. Yeah. So in binary fission, I'll talk about binary fission. So in the binary fission, the organisms which are involved in it, let's discuss that. Organisms involved are amoeba, paramecium, euglena. What are all these? These all are altogether protists. Protists are there. In eukaryotic cells, we have mitochondria taking place in it and chloroplast. Remember what these two are? These two are nothing but the semi-autonomous organelles. So these are also there. Remember this. Now what happens in binary fission? So in binary fission, there are, here I'll go, yeah. So in binary fission, basically first thing which takes place is karyokinesis, then cytokinesis. Karyokinesis is first that nucleus will divide, and cytokinesis that the cytoplasm will further divide, and always two daughter cells are formed at the last. Okay, and remember one thing more, that in fission, whether it's binary fission or multiple fission, parent cell does not survive anymore. It keeps dividing, no? Parent cell doesn't exist. Once after division, the parent cell will not exist. This is very, very important. And we have two types of this division. I told you about two types of binary fission divisions, right? What are those two types? Okay, first is your transverse, second is your longitudinal. So transverse, let's say an example I'll take of transverse. I'll take a protozoa, that's paramecium. In paramecium it happens. Paramecium is what? It is a protist and it is a slipper shaped protist. Let's say like this. And in paramecium, here we have an oral groove from where it takes in food. Oral groove. Okay. Now look. The paramecium, it will divide in a way like, I'll show you. Paramecium will divide in this way. Okay. How it will divide? It will divide in this way. One part is this, one part is this, a horizontal line. But longitudinal, I'll take the example of euglena. Okay, euglena, it looks something like this, no? Now what euglena will do, it will divide in this way. What is this? It's a vertical line, right? So in a vertical way, it is going to divide, and here it is going to divide in a horizontal way. Now remember Leishmania. There is something called Leishmania. Leishmania is an organism. It also divides transversely. Leishmania looks like this. Okay. It will divide, of course, in a horizontal way. So Leishmania also divides in this way. This is the nucleus over here of the Leishmania. Alright. This was about transverse and longitudinal binary fission. Now let's just see what is given here. I'll read it off. Then we'll come to multiple fission. Now look here. It is saying, from here let's see, that prokaryotes like bacteria is there, paramecium, euglena, and eukaryotic organelles like mitochondria and chloroplast. They perform asexual reproduction by binary fission. And in this cells, what happened? The parent cell, it divides to form two similar daughter cells. Remember, similar daughter cells. Okay. And the binary fission, it occurs either by mitosis or by amitosis. The same thing is there, slight difference is there. Now the axis of fission is different, right? Let's talk about amoeba. Amoeba is a protist, right? But amoeba doesn't have any kind of particular definite shape, right? Let's take amoeba. Now amoeba can divide this way, can this way, and this way, and this way. No particular axis is there, right? It has no particular axis because lack of specific shape. Hence, it will divide by which process? Amoeba is going to divide by simple binary fission, right? Now I talk about paramecium. I told you a horizontal line cuts through. It's a transverse binary fission. Whereas if I talk about euglena, okay, that will divide by longitudinal binary fission. That's a vertical line. Now binary fission, it is usually performed by the living organisms during favorable conditions. I told you, when abundant availability of food material is there, they will reproduce in that way. Okay, these were the things which were discussed in asexual reproduction through the binary fission method. Now we will come to multiple fission. Alright, so again I'll come to the board. First I'll explain you, then we'll read it, right? That becomes clear for us. I'll use another board this time. Is it required? No, na? You all can take a screenshot, or you all can also make notes with me. Because during certain times it becomes not possible for me to tell you everything at times. Right, I'll use this part. Yeah. So now we'll talk about multiple fission. Okay. Multiple fission. Now, multiple fission takes place during unfavorable conditions. Unfavorable conditions during many, when adverse conditions are there, so that time multiple fission rate takes place. Okay, and what happens is, let's take an example of amoeba. Okay, let's say this is amoeba. It has no definite shape, no, right? It has no definite shape here in this case. So what will happen, that during this, the amoeba, what it is going to do, it will block its movement of the pseudopodia. It has pseudopodia, right, from where it engulfs the food materials. But during unfavorable conditions, the pseudopodia, it will stop functioning. This is the main part, okay? The pseudopodia, it will stop functioning. And what will happen once the pseudopodia, wait a minute. Yeah, I'll just draw it in a smaller way. Let's say this is amoeba. Now, when the pseudopodia will stop functioning, it will take a certain rounded shape. Okay, when it will take a certain rounded shape, here is a nucleus, no? And then a layer will form, and that layer is the capsule. A layer of capsule or cyst will form around the bacteria, and inside that, okay, pseudopodia movement will be blocked. Now inside that one nucleus was there, it will start dividing repeatedly. Okay, it will divide repeatedly. It will form many other nucleus of the same time. And when the favorable conditions return, then this cyst is going to rupture on all these nuclei. They are actually we call it amoebulae. Okay, this is many other amoeba, more smaller amoebas are formed. This is amoebulae. Okay, let's start dividing. Amoebulae are formed, and these amoebulae, they will rush out. This is an example of multiple fission. Now it happens in our RBC cells also. How? So this was about, I told you about, the amoeba. Now in our RBCs, let's say this is the RBC. So when plasmodium comes, what is plasmodium? Plasmodium is a carrier of malaria, right? So when plasmodium comes inside, let's see what happens. Plasmodium, it repeatedly divides inside our RBC, and then it further ruptures our RBC and it rushes. Okay, it replicates and then it rushes. So this is an example of multiple fission. Alright, I'll erase it for the use. Now we'll come back to here. Let's see what's given here. So you see now, Multiple fission. So here asexual reproduction is performed by amoeba and other similar protists. It stops the formation of pseudopodia. I told you pseudopodia, it is blocked. Movements are lagged. Now see, when the pseudopodia blockage will be there, so amoeba will not be able to move. So its movements will also be blocked. And whenever there is a lack of food, or in adverse conditions, so it will become rounded. It will have a certain rounded shape. It will form a protective covering around its plasma membrane or the plasma lemma, that is cyst or capsule. Now many nuclei, they are formed by the repeated divisions inside the cyst. And after that, I told you, karyokinesis, cytokinesis. Then the cytoplasmic divisions will be formed and amoebulae will be formed. I told you in amoeba, amoebulae will be formed. And that will remain encysted until the favorable conditions will return. Until the favorable conditions will return. When the favorable conditions will return, cyst will rupture, it will break open, and the amoebulae, it will be released. So, through this diagram, it will be clear. Okay, look here. In this diagram, you can see this is the parent amoeba. The division of nucleus takes place, right? Inside this, here we have a cyst. So many are formed, no? And after that, this cyst is rupturing. All are going out. Now, does the parent cell exist after asexual reproduction fission? So, absolutely no. Because if you see, this is rupturing, no? The daughter cells are then binary fission also, like see, this is there. It will divide, it will form two. So, is this existing? No, it is further dividing, it is forming two other daughter cells. So, no, it will not sustain during the fission process. Now, we have budding. Budding in yeast, I don't need to use the board here. I'll tell you only. So, see. Basically, in unicellular organisms, the budding will take place in the yeast. It will take place in the yeast. Now, what do we need to do here? You see, the yeast, how does it divide? Basically, what happens is, it develops an outgrowth. A small bud is there. Okay, it can, it's a unicellular fungus, right? It will produce two daughter cells by mitotic division. Remember, in asexual, it is always mitotic because no formation of gametes is there, right? And this yeast cell is called a parent cell. Now, this is the parent yeast cell. A bulge will appear. This is a bulge, right? Further, it will form a bud here in this case. And then, what will happen? The parent yeast, it will supply the nutrients to the bud. And after the bud is mature enough to go out, live independently, it will come out, and then after sufficient growth, the bud will separate, it will live independently as a daughter yeast cell. Now you see, in budding, let's say this is a parent one. Now here is the child. Now when it comes here, so parent is here, child is here, right? Child is living as an independent daughter cell. So parent is also surviving in this case. Remember, in fission it was not the same, right? Parent was dividing again and again. But here the parent cell is as it is. It is here only, right? It is not. It does not mean that it does not exist. It exists. All right. This was about budding in unicellular. Now we'll come to the asexual reproduction in multicellular. Right now we were dealing with unicellular. Now we come to multicellular. Okay, so telling you about the fragmentation now. Spirogyra. Spirogyra is an algae. Now what happens in this case, basically, the body of that Spirogyra, it will divide into many fragments and spores. Spores called the cycon. Okay. Basically the cycon is written, no, cycon is a spore or sponge, whatever you call it. Now see, in this, the body of the parent cell, it will divide into many fragments. Okay, and each fragment that will live as an independent cell. Whenever there is plenty of water, plenty of nutrients are there, which are available to the Spirogyra, those filaments are present inside, no? So these filaments, they will start to grow up very fast. They'll break into small fragments and they'll live as a Spirogyra fiber. Now, if the body of cycon is a spore or sponge, it accidentally breaks up. So each fragment then will, develop into a new cycon. Clear? Coming back here, regeneration. So, I'll tell you first regeneration over the board. Not many things I have to discuss in regeneration, okay? But see here. Yeah. So, you must have seen lizards, right? What do lizards do? they lose their tail part, right? Well, that is a limited, on a limited scale. Another thing, let's talk about asexually what happens. So remember that whenever we talk about regeneration, it happens in totipotent cells. Okay, funny name, right? Totipotent cell. Totipotent cells are such cells which have ability to regenerate their lost part. Regenerate their lost part. So what is happening here basically in regeneration? What happens, that the lost part, it starts to develop. It takes place in planaria, in starfish. Okay, in starfish and planaria. When the one part is lost, let's say this is a starfish. Okay, let's say one part is lost. So let's say this part was lost. So now this will again form a new starfish. This happens. Okay, the lost part, it starts to develop in totipotent cells. Totipotent cells means the cells which have the ability to regenerate their lost part. That's it. Look here. So you may know that a wall lizard, it breaks up, discards some of its part, tail in emergency. Now that discarded part is regenerated. This is limited regeneration. I told you, this is limited regeneration. Now, however, under certain situations, what will happen, that an animal planaria, okay, planaria, I told you, planaria looks like this. Animal planaria, let's say it divided. Now this again will develop into a new planaria. Let's say it broke up its body into two parts. Thereafter that each part it will regenerate and it will form two new planaria. That's what we call as regeneration. Now let's come to budding here. Okay. What I'll do today, I'll do till spore formation. Sexual one I'll discuss in the other video. Otherwise it will be very long. Okay. Look here in budding. Alright. So in case of hydra, budding in multicellular in hydra, what will happen, that under favorable conditions, specific part of it's similar to yeast, okay, just that it's a multicellular one. Okay, what will happen, a bud will, outgrowth will come out, the dermal layers, their continuity with those of parent hydra will support the nutrients and all. And the budding hydra, it will separate from the parent hydra, start to live an independent life. That's it, nothing much. Okay, this is the nascent hydra, which means this is, one second, yeah, nascent hydra means this was the hydra which developed from the bud. So remember, nascent hydra is a hydra which develops from the bud. Clear? You see here, I'll tell you. For example, you see here, this is bud formation took place and outgrowth came, and then that outgrowth developed into a hydra. It's a small younger one, so we call it a nascent hydra. Now vegetative propagation. Remember that vegetative propagation only in plants. Okay, only in plants, not in any organism, only in plants, means kingdom Plantae. It takes place only in the kingdom Plantae. Now in this, what will happen, the reproduction will not take place through any reproductive part of the plant. In this type of reproduction, the reproduction is going to take place in the part of the plant which does not perform any reproduction. Like which plants? Like which parts? Like the parts of stem, like the parts of leaves, like the parts of the buds and roots. That is when we call it as a vegetative propagation. Now in potatoes, what happened? Small buds appear, That is what we call as eye bud in a potato. In a potato tuber, eye buds appear. So those eye buds, then they develop into new potatoes. Clear? That is through the stem. Potato is the stem, And if you see a leaf, so bryophyllum, new leaves appear on its leaves only. New bryophyllums appear on its leaves only. So that's an example of leaf margin. On the leaf margin it occurs. If you talk about sugarcane and grasses, it will happen with the help of on the nodes. Okay, with the help of the buds which are on the nodes. Now in sweet potato, it happens through the root. Okay, it happens through the roots. The sweet potato roots. That's the vegetative propagation. Clear? Now let's come to spore formation. So Spore formation, it will take place most often in fungus. You'll see now when fungus attacks, it forms a greenish kind of a layer. Right? How does it form it? Let's understand that. Let's say we have a fungus like rhizopus, or let's say mucor. Mucor, they have filamentous body. This is the body of a fungus. This is a rhizoid. Many bunches are there. That's the hyphae. Now this particular, if I just zoom it, this is the sporangium. Inside that many spores are formed. Okay, spores are formed. And when the sporangium, it ruptures, so these spores come out. Alright, these spores will come out, and they, they need a moist climate. So whenever they see that a humid climate is there, that place humid, it's a wet place, so these spores will start to germinate there. They're just like the seeds. Okay, when the sporangium will burst, these spores will find a place to germinate, and there they'll germinate and form a greenish structure. That's it. Okay, this is the same thing which is written. They have sporangia. I told you sporangia, and inside the sporangia spores are formed. Sporangia breaks, spores are released, and spores, they'll find a warm and moist place, and there they'll form a colony which is greenish in color. Okay, so this was about asexual reproduction in in unicellular and multicellular organisms. Now in the next one, I'm going to do sexual reproduction in plants. Both I'll discuss the pollination also, only in plants. Then we'll come to human beings. It's a very interesting topic. I hope no doubt you're not left with any kind of doubt in here. So yeah, all right, this was it with what we did today. So here we did asexual part. This is completed. Now this one divided into two categories: plants and animals. Plants one I'm going to do in the next part. Alright, so thank you everybody. Let's wait. Let's see what happens next. Okay, thank you everyone.